प्रेजेंट नाउ का एरो करेंगे, उसके बाद आएगा विंडो में इंटायर स्क्रीन का, तो वो करेंगे। फिर आप जो भी स्क्रीन पर शो करेंगे वहां इंटायर स्क्रीन के बाद फिर एक विंडो आएगा फोटो वाला, उसको क्लिक करेंगे।
प्रेजेंट नाउ,
हाँ प्रेजेंट नाउ करेंगे तो फिर आएगा एक पॉपअप, वहां आएगा इंटायर स्क्रीन। इंटायर स्क्रीन पर क्लिक करेंगे, आया।
वो सफारी के सेटिंग में कह रहा है कि वो मेरा मैक है ना, उसको सेटिंग में करना पड़ेगा। मैं एक सेकंड दे दीजिए, उसको सेटिंग में कर देता। सेटिंग में नहीं है मेरे, बट इस मैंने कई बार प्रेजेंट तो आई हैव ऑलरेडी प्रेजेंटेड सेवरल टाइम्स।
गूगल मीट में किया ना?
हाँ
वही तो, ऐसा तो नहीं होना चाहिए सेटिंग कुछ।
लेट मी करेक्ट, जस्ट वेट। Myths and apprehensions. So many people talk about these GM crops. The first myth is that when we talk about this, not natural. So let me start the entire journey, that how everything has started during the Green Revolution. You all know that India had the scarcity of the food grains. Long Green Revolution in India. So you all know that recently Dr. M.S. Swaminathan has been given Bharat Ratna, and he was also one of the pioneers in this particular field, and he was also instrumental in the Green Revolution in India. So this person, this Norman Borlaug, he is also known as a father of the Green Revolution, and he has also got Nobel Prize, in all these things. And for the interest to the students and the participants, that he has not got the Nobel Prize in Science. He has got Nobel Prize in Peace. So if you have a full stomach, then probably it will be easier to feel yourself happy, and you will not fight with each other. So that's why he has been given this Nobel Peace Prize. As well as if the participant, they can talk to their grandparents or their parents also, that in 60s that time Prime Minister Lal Bahadur Shastri, he has urged the entire nation to at least do 1 day fast, because we were having the grain shortage. From this, that time population was also less, but even the population is so much has got increased, but also we are able to feed the masses. And the main, there are so many things are there. So we will just go hurriedly to what has happened during the Green Revolution, some good thing and also some bad things. Because of the fact that now we are looking forward to 2050, when we have to feed a much larger population that is going to be in India and entire world. So the food security is the primary concern. So there should be another green revolution that we have to start. So before that, if we know how this first green revolution has been taking place, then we can go for the next green revolution. And the mistakes what we have done during the first green revolution, that also we have to consider in our the next. So basically our idea is that you have to go for a sustainable type of agriculture development, not just the general type of agriculture development. So you can just see that the green revolution, usually when we talk about green revolution, it has happened mostly in India and Pakistan, and there you can just see that these are some of the growth of the earlier crops were there, and actually some of the crops which were growing already in the Mexico, So Dr. Borlaug was from the Schmidt Institute, and he has the Schmidt Institute is in the Mexico, and there he came and he has taken some the varieties which were disease resistant, semi-dwarf varieties, and turning country to grain importers to grain exporters. So these are some of the success story. So the strategy what has adopted during the Green Revolution is the use of high yielding variety, mainly by breeding that they have done. The Indian varieties were long and they were disease resistant, but they were low yielding. However, the Mexican variety were disease susceptible, and the Mexican variety was short in height, and it was having more green, the per hectare that people used to get harvested. So that's why that they have crossed with the Indian variety, and they wanted to retain the disease resistant the character of the wheat and the rice, and they also want to integrate what we call in this particular term is that the integration to the variety which you are developing, that should be the high yielding. So the first thing is that they use the high yielding varieties. They also use the chemical fertilizers and pesticides. This I will discuss a little bit more. These are basically some of the problem now with the Green Revolution. And then new projects were built, irrigations were potential were generated. Use of tractors, motor pumps, and farm machineries got increased, and also during this time, the nationalization of bank has taken place, and the bank started lending the agricultural loans to the farmers. All these are the important thing for the Green Revolution. And impact of the Green Revolution is that the Green Revolution in agriculture had increased the yield of the food crops in rice and wheat. Besides that, other crops has also got the improved, and they also got the high yield of many other crop plants. The nation attained self-sufficiency in the food production, and government of India has started maintaining the buffer stock. Now that time the FCI, the Food Corporation of India, has been established in 1964 for the food fertilizers and seeds, and they used to store in the stores of FCI godowns. And then the government were able to curb the inflationary trend of the food product. So the important thing that I have already mentioned, that environmental impact on Green Revolution, that we have to take in the next Green Revolution, in my opinion. So what has happened, like if you see that the per capita the consumption of the pesticide and insecticides, you can just see that insecticide in India is quite high, is very, very high, and the world is still less. So you can just see that all the year excuse me. So all the years that you can just see here, the 1955 to 56, the per capita the consumption of the pesticides and fungicides was very, very less, almost zero. But you can just see that these 74, 89, 92, you can just see that these are the period where the Green Revolution has taken place. So there it has just shoot up this pesticides and fungicide things, and still now people are much more aware. It has gone down, but not to the range where the 1955-56 was there. So that means it's very difficult to understand all these things. Like here you can just see that the per hectare fertilizer used in the states of India is here you can just see that Punjab is the highest. And you all know that Punjab and Haryana was the two states where most of the Green Revolution started, and later on it has got spread into other part of our country. So you can just see that it is much more higher than the the Indian things. And you can just observe in recent times that if you all are from the villages, you just go to your villages, the ponds are now turning green. And that is because of the algal growth. Algal growth is being, has a boon because of the fact that the fertilizer which has been used into the fields, they are getting leached out, and they are getting collected in the ponds, which is, the phosphorus fertilizers are more, and that is also, is a major cause of the increase in the algal growth in the ponds and the small rivers. So you can just see that, you can just Google this things, the cancer train. So just go, this was a shocking tale of the India's cancer train. And this cancer train, you can just see that it people used to call from, there was a place in Punjab that is called as Bhatinda, and the cancer hospital was in the Bikaner. So there was a train which used to start from the Bhatinda to Bikaner. That was an overnight train, and most of the passengers in the train, either they are the cancer patients, or they are attendant that they used to travel. So that's why they called as a cancer train. If Google, you'll find all this information. So that means the increase of all these cancer and everything is very, very difficult. That what they can understand. There, you will just go to. I am also working on one of the major disease of tea. And recently, I visited to some of the tea gardens, where I found that they are using liquid copper fungicides for the control of a disease which is important for the tea production, that is the blister blight. And because of the commercial pressure, usually you have to pluck the leaves only after the 7 days of the spray. But because of the commercial pressure, they used to pluck to second day or third day also. And that actually what we are taking in our cup of tea is actually some part of these copper fungicides. So all these things has started, with good things has come, but some with the good thing, some bad thing has also come. So all these things, why I am telling to all of you that in the second green revolution, we have to understand that these are very, very bad for the environment, the human health. So we have to think about all these things and go beyond all these things. And the most important thing what we found is that if you want to have a disease-resistant plant or the pest-resistant plant, the transgenic way or genome editing way is the best way to curb all these diseases and get good yield. So these are some of the challenges of the green revolution in the 21st century. That the saturation of the crop improvement opportunity that has happened, like whatever we wanted to get the increase of the yield is already we have achieved. But we have to, you all know that most of the farmlands are now getting, because of the fast urbanization, it is now wherever you found, like 10 years back, the field was growing, the wheat and rice or any other plants. Now, if you move to those areas, you will find the buildings. So that means, the agricultural land is shrinking. So we need to have, it's very important to improve our crop beyond the breeding strategy. So that's why, the sustainable agriculture and the environmental perspective is also very, very important. And that's why I have given in my, introductory slides some of the slides about the cancer traits and other things. And then speed of improving crop for the better yield and value addition, that is also very, very important nowadays. So we will discuss about all these points in detail. So if you see that the per capita availability of food grains was very, very less in 1951, but 2014 It is near about 324. It has not increased much. Now recently I found a data where it is near about 348. So it has not got increased that much. So our population is increasing in geometrical ratio, and where the food production is arithmetic ratio. So that's why we need to have a second green revolution in our country. And how the most important challenge is that how do we feed more people without further damaging our planet? This is very important. So recently, I think 2016 or 17, our Prime Minister, he has called for the second green revolution. And the second green revolution can be only possible by different methods. We will be discussing about those things, that how we will go for the second green revolution. Have we learned from our mistakes of the first? GM plants or genome editing plants will be helpful. Myths and apprehensions about GMOs, which is the most important things that we will discuss today. And are we serious about the doubling of the farmers' income by 2022? Still, we have not achieved that part, in 2024. And public perceptions and acceptance of the technology. And always, I am very positive person, and I always talk about the way forward in the end of my talk. So GM crops and genome edited crops, most of the things which we do have the apprehensions are they are not different from the naturally occurring plants. So first we have to understand what are the naturally occurring plants. Then we will be understanding that कभी भी बात करोगे तो they will be talking about all these things कि ये तो natural नहीं है, ये तो बाहर से आप introduce कर रहे हो. अभी आपको when I will discussing about some of my slides, you will come to know that whatever we are eating nowadays, nothing is natural. It has already modified and several times, and the first modification has taken place near about 10,000 years back. So we will talk about that, how the people have started modification in long back, and they are also going to change the entire gene pool. This is the first thing that people do have in their mind, that they are going to change the entire gene pool. They are bad for human and animal health. जब कुछ ये हम इस तरह का ट्रांसजेनिक प्लांट खाएंगे तो they are bad for the human and animal health, and they are bad for the environment. In my opinion, this is not as bad as it was earlier in the first green revolution, when the pesticides and fungicides has taken place of all those the organic fertilizers, and the people used to go in the field with cow dung and other things long back. But now, because of the fertilizers, that it's also not the use of the fertilizers, the indiscriminate use of the fertilizer and pesticide, fungicide. What has happened, like whenever a farmer goes, if you are not very much literate, even though you are literate, so how they come to know that which the pesticide they have to spray, and how much the quantity that they have to spray. So usually they ask their shopkeepers in their villages, or the kasbah और जो पास में शहर होता है उसमें जाके पूछते हैं. And then they usually those shopkeepers, because they want to sell more, so वो suppose they have to use that only 5 ppm or 2 ppm, that is part per million. So they will be giving you the more so that it will be sold more. And the important thing is that sometimes it happens, like it for the fertilizers also, that you go to the fertilizer shop, you ask that कौन सा अभी अच्छा चल रहा है, तो नहीं ये वाला fertilizer सब लोग ले जा रहे हैं. Despite of you all know that in fertilizers, nitrogen, phosphorus, and sulfur, the NPK, potassium, they are very, very important for the soil health. And because of that, suppose the phosphorus is less, then only you require to have phosphorus. Otherwise, if the nitrogen is less, then we will require to have nitrogen. But they will go to just, or बगल वाला फील्ड वाला क्या हमारा स्प्रे कर रहा है, कौन सा चीज ये कर रहा है, उसका ठीक हो गया हम भी डाल लेते हैं. So similar type of things they talk about, and sometimes they get some ideas from the relatives also जो दूसरे गांव में भी रहते हैं कि वहां पर हमने ये छिड़का, आप भी ये करो छिड़का. So this is basically not the actual correct use. That's why the Prime Minister has already started the soil health card, and the many people who are involved in the Krishi the Kendra, Krishi Vikas Kendra, and they are actually doing this type of soil health card, and these are very, very important for the agriculture development. So that we will discuss about. So first thing that we will talk about the myth of natural food. The food that we eat comes from the plant already extensively modified from their original form, and even the heritage varieties are extensively and genetically modified. Let me prove all these things, how this is happening. So it is not modified recently. It is modified, for thousands of years. Like we do have the certain, you all know that usually that the human population was originally, that they were nomads. मतलब एक जगह वो टिकते नहीं थे, वो इधर से उधर जाते रहते थे. So they used to, when you all know that most of the civilization has developed in the river basin. जो रिवर का जो नदी का बेसिन होता था वहां पर सिविलाइजेशन डेवलप हुआ. वो किस लिए हुआ? बिकॉज दे वर गुड फॉर द एग्रीकल्चर, एंड फॉर द एग्रीकल्चर you have to sow the seeds, and then you have to wait for 3, 4, 5 months for the harvest. So this type of things what will happen, like you have to stay for to that particular area. Then it all started the colonization and the development of different type of the colonies. Even the Harappan and recently the India, the Rakhi Gadi excavation has already proven that even maize and some of the things, barley was was already grown in the fields during those era. And they are actually major crops that were domesticated. So just try to understand what we mean about domestication. So we will be discussing about these things. I will be giving you 1 or 2 examples. These are very simple examples, and you will just understand that. I have already discussed that most of the civilization started, in the river basin, and here, in India, these are the archaeological records that in near about 8,500 years. The rice, beans, and even all those plants, even chickpea has been evolved in the Indian subcontinent. Other part is also the corn, the squash, beans, and potato in the Southern America, just like that. The China is also the rice. These all are the archaeological evidence of the excavation. So you can just see that how I told you that it is important. What is selection? What is the domestication? So what has been like natural variations? They occurs everywhere in the plant system. Like here you can just see that out of these, which one you will like to take? This one, because this one is having the more grains. Here also you can just see that out of 1 or 2, 3, 4, 5, you want to take only this, and they just take the seeds and store the seeds for the next generation. The next generation again you are going to sow the seeds. Then again you are going to do this type of thing. So this is how our forefather has actually done this the domestication. And I do have some of the archaeological evidences near about 7,000 years back. The bhutta jo tha na aapka maize, it was very, very small. So and after the domestication, near about 500 years ago, you can just see that how this the crop size has already got increased. And this is the archaeological sites excavation in Mexico, what people have found. So that means people have already started modifying these plants by this is we call as a now in modern terms we call as a selection breeding. So that our forefathers have already done in these things, and this is one of the examples. The teosinte is the original maize. It was present, and it was much more branched, and also it was having a covering. Here you can just see that. So if there is a covering over the seeds, then it will be difficult to harvest. So which type of things that we got selected, that it should not have any covering, and it should have just one erect the plant, no branching, so that you'll have a better yield in this case. So how this maize has already got so this is teosinte. This is a modern corn which has already done. So this is one of the examples of the one of the rice variety which has been developed during the first green revolution. That is IR64 rice variety, and this is the usually what has happened, like in IR64, the original rice was crossed with other with the Mexican varieties, and after the crossing of the variety, you can just see that this is in blue color. The mutations are some naturally occurring mutations were there. Some translocation inversions were there, and then there will be a crossing. And after the crossing, whatever the original rice was there, so there was a crossing, and you can just see that the so many modification already has done in the case of IR64. So this is even though you are doing the normal breeding by crossing, this is important nowadays. Is very, very important nowadays to understand that it is already being modified, several times. So the take-home lesson is that nothing is natural. It has been, modified, since ages. So the new advances in the genetic technology, which can improve further the improved variety. One we called as a marker-assisted selection. This is we have already started. I have got a big project on this marker-assisted selection on the breeding of chickpea, and which I am doing with Indian Agriculture Research Institute. So this is important, this is much more advanced than what people have done, the MS Swaminathan and Norman Borlaug. And this is in those variety the interrogation, they used to cross and back cross, we call back crossing. They used to go for near about 7 to 10 generations, sometimes more generations. But if you want to do the marker-assisted selection, the interrogation of the gene you can find out, or you can fix in 2-3 generations only. And just after the segregation, you can figure out they are homozygous, heterozygous, or any whether they do have the character which we are looking. Those things, even without destroying the plants, you can know by this marker-assisted selection. Then the genome widing association studies now is very important, and we will be discussing a little bit about these things if time permits. And then the recombinant technologies and transgenic plants. Then we call about, we will discuss little about cisgenics and intergenics, intragenics and transgrafting, and precision genome editing. And this I will just touch all those things, because my idea is that we have to understand the basic myths and apprehensions in this particular things. So usually in the first green revolution, what people used to have in the selection, in the crossing breeding by the crossing, that if you have an elite tomato which is having the high yielding, but this was disease susceptible, and poor tomato, which was a small tomato, but this was disease resistant. So what do you want? That you want this character which that is given in yellow, that is a disease resistant character, to be integrated in elite tomato, which is which you can do by crossing. And you want to retain all the characters of the elite tomato, only the character which is important for the disease resistance, that you wish to get integrated. So what has happened, like whenever you are doing crossing, it is not that only this will go precisely. So beside that, what will happen, that when you are going for the crossing of the two varieties, it happens like some part of this, the poor tomato also comes into these things. So this is the drawback of the breeding, which we can definitely you can get rid of all these things by the transgenic technology or genome editing technology. So, there's one example also that you can just see that this is a submergence tolerance rice, which has usually this rice variety, which was low yielding, but it can survive in the floods as long as the 17 days. So when the water recedes after the flooding, the normal plant actually, when water retreats, it actually dies, but the submergence tolerance remains erect. So this people have identified this particular rice variety from Odisha. Some people claim that it is from somewhere near Bangalore, and it doesn't matter. But submergence sub-1 rice is already known, and if you cross through both of the things, you'll get this is the one of the example. This sub-1 gene is in yellow, and Swarna was the elite variety which was growing is the rice variety that is in orange color. And you can just see that after the crossing, what happened, it is having near about 50% of this the gray and 50% of this orange. Besides that, your sub-1 gene is also in yellow. It is there. So what has happened, you keep on crossing with this Swarna variety that we call as back cross, and then probably you will get a product where the Swarna variety is there and submergence tolerance gene is also there. So that means even though you are doing with breeding, it is already you are modifying the genomic things in most of the times. You can just see the result of that. This is Swarna sub-1 and this is Swarna, and when after the water recedes, this Swarna sub-1 it doesn't die, and this is, actually gets die, which is the normal sort of variety is there. So there are several advancement in the breeding technology which has started, and the genetic modification GM crops is another breeding method. What we call nowadays is another breeding method. So the difference is that earlier it was the tomato with the tomato variety. So rice with the rice variety. Here, if the disease resistant the gene is present in the Rhodopsis thaliana, you can take it by genetic engineering. You can just take out the gene and transform to the elite tomato variety, and you can get a GM plant which is resistant to the particular disease. So it's very easy process. As well as whatever the modification there, lots of modification here. Precisely, you are doing the modification of the things. So there are different methods are there, like molecular breedings that we are right now we are doing. GM is also one of the method, and some of the plants which can be propagated vegetatively. Also, we want to go for the precise molecular breeding. Another thing we call as cisgenics. Whenever we talk about the GM crops, we talk about one thing most of the times, that is the BT toxin. So BT is not all about your GM crops. So it is beyond that. Suppose that if one variety of apple you are isolating a gene and transferring it into the another variety of apple, it should not be people should not get afraid of these things, because with this apple gene which is getting transferred to apple gene. So this is called as a cisgenics. So cisgenics should be allowed in my opinion, and several type of the cisgenics people have already started, and they do have several examples of GM crops are there. I just want to go hurriedly to all these things, like this BT toxin that you all talked about. And another example is the banana via pepper gene. The pepper, they have isolated a gene which was against the a banana bacterial wilt disease, and these people have got. And that this because of that, there was one Indian scientist, Dr. Leena, was involved in this particular work, and this has been done in the Congo. And some of the West African countries, where the banana was a staple crop, and because of this particular disease, their economy was very, very bad, and it has spread throughout their fields. So they were trying to save all these things, and that scientist, the Indian scientist, he has got highest civilian awards of some of those countries. Just because of that, because she has got these resistant plants. So there are other examples are herbicide resistant plant. So vitamin A is also like golden rice, golden cauliflower, golden maize. All these things that people have already started. During the COVID time, people have already started working on the expressing antigen of or the spike protein of the COVID-19 into the transgenic plants, antibodies, many other things people have started. Biofortification of the plants also, like some of the plants which are the rich in starch and carbohydrate, they wanted to have some more protein into the those plants, so that people will be getting some part of the protein in their diet. So such type of things people have started breeding for the drought tolerance. These are some of the examples of the drought tolerance. So the prepaid crops for the green revolution is also important. Like transgrafting also I have talked about. Like suppose one disease is important for which is actually infecting the plant root. So what you can do is that you can have a root system which is transgenic, and after that you can just graft your upper part of the plant to this plant, so that the plant roots will become resistant to the disease, and the other part will not be having any effect on it. So such type of things people have already started. You all must have heard about recently the different we call as a different techniques which we call as a genome editing technique. So recently, the two scientists has got a Nobel Prize on this genome engineering, which has started from the plant system. So not from the animal system, from the plant system they have started. So all these things are important nowadays. The CRISPR-Cas is now in picture, and I'm not going into detail of all these things. So I can give a lecture on these things also in details, but I don't want to go for that. But I will discuss about much more important issue, which is important for the acceptance of these the transgenic plants. So the important thing is that our GM crops are safe to eat. I tell that it is yes. Most of the time it has happened like the people do have the data कि मेरी बकरी जो है वो खाई ये प्लांट और वो मर गई, है ना. तो मैंने उसको ये टमाटर जो ट्रांसजेनिक टमाटर खाया और मेरे को हेडेक होने लगा. हो सकता है वो हेडेक जो है वो अपने वाइफ से वो पूरा लड़ाई किया होगा और उससे उसको हेडेक हो गया होगा, या बकरी को खुद ही मार के खा गया होगा. तो यू नेवर नो. So what has happened, like important thing is that that is just a joke apart, but the important thing is that when you talk about these GM crops, it's also important to have a good having a good regulation. We do have a good regulation system, and this is also important that whenever you are eating all these things, unknowingly you are eating, if you are eating all these things, and that is going to affect you a lot in all these things. So it's always better to have a regulated things of these GM crops. Second thing is that when we talk about these GM crops, what I was discussing about, like whenever people do have the data, it's just 1 or 2 or 3 incidents like that मैंने खाया मेरे को headache हो गया or like that. So there should be a statistical significance of all those things, in my opinion, so that you can get a very good idea about whether this is bad or good for your health. So you can just see that if you go to your market nowadays, you will see that the papayas are now turning. You will get the good reddish in color papaya. So earlier, the papaya was not that reddish. It was just, the yellowish in color. But now that papaya is basically a transgenic plant. It has come to India somehow, we don't know, and it is being, adopted by our, population. It is much more sweeter also, and people are growing. Similarly, like if you go to, the many part of your Gujarat and Maharashtra, even Andhra Pradesh, I have visited many places. Even the 90 or more than 90% of the cotton is now the GM cotton or BT cotton. So it's not like many apprehensions were there in people's mind that we all have to present, take the seeds all the time from the company people. So what about the people who are developing these transgenic plants from the university system or institutes? So in a government institute, people are developing. People should accept all those things. There's nothing called, that we are going to have, this type of seeds only for that. Earlier also for the breeding, you have to go for the good seeds. You have to go all the time to the market. Similar types of things that you have to go for all these things. If you are from the scientific background, usually what has happened, like there is a the homozygous and heterozygous, and there is a segregation. So it's always better to have the plants which are good in health, and that you can take from the market. So there are many people, like you can just see that this lady, you all know that Vandana Shiva, and then Jeffrey Smith. Jeffrey Smith has already, he was most of the time he was very much against of the GM crops. Even this lady Vandana Shiva, and they were actually not very much against to the transgenic plants, but they were actually against to the multinational companies. They believe that multinational companies are going to, affect your farming and other things, and which has already, given a bad shape in acceptance of this. The people are the scientists like me. They are not that much vocal. These people are vocal. You just listen to their talk. They will be, all the time they will be talking. They are shouting. They were all talking about all this nonsense, and people used to accept all those things very difficultly. I can tell you that one example. Even the breeding plants in the first revolution, when people have got that time also, it was a problem to acceptance of all these things. Sometimes I used to talk to my nanaji or my maternal grandfather. He used to talk, इसमें स्वाद नहीं आता है, इसमें ये नहीं आता है, ये अच्छा नहीं है, इससे कैंसर हो जाएगा, इससे फलाना हो जाएगा. So all these type of apprehension, and these people are nowadays, they are spreading all these things a lot. So worldwide, I think 3 or 4 days back, I have listened to one. I have got one news from China that China has approved near about 81 maize variety in China to improve the maize production in China. You can understand that in the Bangladesh, even the transgenic plants are transgenic brinjal, and the BT brinjal is already in the market. And somehow it has entered last 3 years some places in the eastern part of Bihar and Bengal. And the people are already started growing transgenic brinjal. Somehow it has got imported from Bangladesh, and people are starting growing. So it's always better to have a very good, the monitoring system besides all these things. So this was one of the news earlier. India nears approval of first GM fruit crops. That is in 2017 by that time by Chancellor of Delhi University, Professor Deepak Pintal. But still, it has not got finalized. And this was one of the things. Like you can just see that even Maharashtra farmers, even the Shetkari Sangathan, they have started, fighting for the GM crops. Now it is called as a Rafales for Jawan. Why not Jugaad? Why Jugaad for Kisan? Let us sow GM seeds. So all these things, people have already started talking about all these things. family of the facing GM brinjal storm. So this is also one of the important news that we have got, that in the Fatehabad this news has come, and somehow this GM brinjal has started, people started growing from somewhere. So there is in my opinion, there is some concern about the biosafety, and in the Indian scenario, we have to have some societal concerns also, like unfamiliarity with the technology, lack of reliable information, negative media opinion, opposition by activist group, and mistrust of the industry. So it has happened, many other countries. Most of these people, like Greenpeace and other peoples, are very much active in European countries where they don't have, the problem of the food grains. They can import from anywhere, and they do have this type of activism, and these are very bad for this type of things. Like environmental concern is also there, like horizontal gene transfer, which is a genuine concern, and people who are working on these transgenic plants, they should talk about these in more details about their own products. Effect on non-target organism, that is very unlikely, but definitely you have to before releasing those plants in the field, you need to figure out whether they are important or not, and they do have any impact. And then development of resistance by pest. This is very, very important point, and this also we have to have this type of concern. So there is a regulatory framework of GMOs in India, and there, the Department of Biotechnology, Government of India is having made there is called as a genetically modified crop and rule of manufacture and use, import and export. They have already monitored. Although the bill for all these things is lying in the parliament since 2003, and still it has not got passed. And although India has already made a genetic engineering appraisal committees, where the ministry, it is under the Ministry of Environment and Forest and Climate Change. And then there are so many committees that you have to go for: district level committees, state level committees, institutional biosafety committees, genetic engineering and approval committee, GSE, then RCGM, that is a review committee for genetic manipulation, and the Government Genetic Advisory Committee. All these things already people have made, and there is a framework by which you can do all these things. That applications should first go to their institutional biosafety committee, then RCGM, then GSE, then considers for environmental release, and then there will be so many, the feedbacks and different field trials should be required in all these cases. These are important cases. So good thing, recently has happened, like on 30th March, the Government of India has passed the genome editing plan. That is only SDN1 and SDN2, that means 1 or 2 nucleotide modification or insertion, that they have identified, and they have found that it is good for the country, and this we welcome, like scientists like me, we welcome all these steps. Maybe this is the first step towards going for the acceptance of the GM crops. So I end my talk with a positive note that if we link public policy, science, public understanding for the bright future of India, we can make a difference, definitely. So thank you very much. If you have any question, you can just ask me.
Thank you very much, sir. So you have started with breeding program, and then GMO and GM, and there are different terminology related to gene modifications.
Yes.
Yes, so now participants, it's open for question answer. If you have any question, any doubt, please ask question. Yes, Hakim Javed, your hand is raised, and I have requested you to write in chat box, but you have not written.
Yeah, it's all right. He can ask me. Yeah,
yes. No, so now you can ask question if you have question. Anupma Kumari is also.
Yes, sir. Good evening, sir. It was very informative interaction, sir. Sir, actually, I am Dr. Anupam Kumari, working as research associate in GVRC Gandhinagar, Gujarat.
Great.
During my PhD, I have developed 3 transgenic cells using salt responsive gene. Sir, I have one general question. According to you, what would be the future of genome edited plants due to GMO regulation? Is it only for
Yeah,
research?
that's why I have, the last second last slide, I have already shown that Government of India has already
Yes, sir.
approved. So it will be, you can, it can go in the market, but there is some regulation. You have
Yes,
to
sir.
get it tested, and then you can go for it. So genome edited crop, where the big chunk of DNA, that is SDN3,
Yes, sir.
that is not allowed yet, but the small modification like 1 or 2 nucleotide, that is allowed.
Yes, sir. Sir, currently I am working on CRISPR-Cas9, gene editing of GBSS in potato.
Very nice,
Sir,
very nice. All the best.
Yes, sir. Thank you, sir. Sir, but we are using selecting markers, sir, kanamycin. Then what do you think it would be?
No, now the genome edited plant, because of the genome editing, you can segregate out. So that you
Yes,
can do.
sir.
So there are so many methods are there. So
Yes,
that is important. So this is also important. Okay.
Yes, sir. Okay, sir. Thank you.
Okay.
Yes, Rakesh ji. Hello, please unmute and speak.
Hakim Javed and Raksha Bhavkar.
Yes, sir.
Raksha ji.
Thank you so much, sir, for very nice lecture. Sir, I just want to know that with GM crops is like we have seen that so many issues. So like we can go for only CRISPR-Cas9 directly like that.
Yeah.
As CRISPR-Cas9 is not being considered as transgenic, right?
Yeah,
sir?
no, two different. I mean, now SDN1 and SDN2. That is just 1 or 2 nucleotide change is not being considered.
So it will be not like exactly totally like GM crop. So like CRISPR-Cas9
Correct.
can
You
be
can
considered as
segregate the selectable markers also, and you can get the even selectable marker, like what the earlier speaker was telling about the kanamycin resistance or something like that. That you can get rid of that.
Yes.
Very easily.
Raksha ji, using CRISPR technology, either you can delete or you can insert gene also. If you are inserting gene from outside, that will be again considered
transgenic.
So that is the SDN3 people have government has still not, where the large chunk of DNA is being inserted, that has not got the approved yet, but at least some beginning is there. The small plants, the small changes that is allowed 1 or 2 nucleotide.
Yes, sometimes single nucleotide change may happen.
Yes. Many people I'm working in this particular field where people are working on now, besides resistant, people are working on susceptibility, which actually facilitate the disease progression. So if you know the susceptibility genes, you can just mutate that susceptibility gene and you get a resistant plant. So such type of things is very easy for getting in this case.
So for legally, like it is more easy to get convinced as compared to GM.
Correct.
But you must have idea about that susceptibility gene or the gene which are responsible for attachment with the pathogen.
Yes, correct.
Pathogen interaction का research हो रहा है ना plant pathogen.
Yes, sir.
How pathogen interact with the receptor of the plant or human or animal, and then they enter. So if you will mutate that, then pathogen may not enter. Am I right, sir?
Sir, yeah, correct.
Hakim ji, you have raised your hand, but you are not asking question. Hakim Javed.
Hello.
Yes,
please ask.
Sir, my question is that can we to consume for this time watermelon or not? There is a controversy in Jammu and Kashmir during the fasting days. That one thing is that people have a suspense that we will to consume more and more watermelon this month. But this time we cannot to consume that. we have a suspense that they are to be added some chemicals like calcium carbide or some we can use the hormones. Sir, what is your opinion about this?
That is for watermelon you don't have to do for all these things. I think these are the afwah hai. So that you don't have to bother about this. Sometimes it happens. Sometimes the shopkeepers also does this type of things. Like the things will be the market will be destroyed sometimes because of that. But here in Delhi, I don't know about the Gujarat, but in Delhi we are today also taken the watermelon just like that. I don't think
गुजरात में भी तो खा रहे हैं सर.
nothing is many times it happens like even the cauliflower is bad. Sometimes all those things are bad.
हकीम जी कश्मीर से बोल रहे हैं.
So yes, don't worry about those. I don't think that there should be any सच्चाई इसमें कुछ भी है ना. So you eat whatever you want.
Thank you, sir.
Any other question from other participants? You can share your feedback also depending if you have learned new things.
Sir, I have one more question.
पूछिए ना.
Sir, if you are using the same gene from same plant and transforming using the Krylok system, sir, it would become in nature or not?
You see, Krylok system earlier people used to have, but now with the genome editing, we don't have to go for the Kryloks. So genome editing can be done very precisely to that particular gene, and there are some issues of off target, which so many the modifications that now people do have, and you can get rid of those off targets. You can also screen the plants before you release for the off targets.
Yes, sir, yes.
So that you can do, but Kryloks I don't think that is important right now. Earlier it was important for the precise insertion of that particular thing. Here you can just design a gDNA based upon the pest sequences and other things, and you can just get it.
Well, sir, actually I am working on both the objectives. I am overexpressing start branching and then using the Krylok system and also doing the knockout of the VSS gene. That's why I asked, sir.
Great. All the best.
Okay, sir. Thank you so
ने
much.
GSM CRA से PhD किया है सर.
अच्छा, good, great, बढ़िया. Any other question?
No, sir. Thank you so much, sir.
Any other participant? Other participants, you may share your feedback, oral feedback also.
कुछ भी question पूछ सकते हो. It may not be related to my, you can ask any question.
You can ask about NIPGR also and GNU also.
Any place, anything.
सर मेरा एक क्वेश्चन है कि सर हमारा फ्यूचर क्या हो सकता है? प्लांट का तो इंडस्ट्रियल लेवल भी नहीं ले रहे हैं.
नहीं, ऐसा नहीं है. फ्यूचर किस चीज में आपको बनाना है उस पर डिपेंड करता है. अगर आप
रिसर्च में हम सर इंडस्ट्री में भी ट्राई करते हैं तो वहाँ लेते नहीं है प्लांट के लिए.
नहीं, क्यों नहीं लेंगे? बहुत सारे जगह पर लेते हैं. अभी बहुत सारे कंपनीज आ रहे हैं, बहुत सारी चीजें हो रही हैं. अब चीजें काफी ज्यादा डेवलप कर रही हैं सब चीजें. You can just hope for the best. Things will are changing. Many companies. You can start your own startup.
Yes, sir. I am just thinking, sir. But I don't have too much knowledge.
She is busy 12 hours in her work at GBRC, I think. She is not having extra time.
What is the institute?
GBRC Biotech Research Center has.
Oh, I think day before yesterday, Sudhir Singh has he joined. So he is known. He is a very good scientist. You can just talk to him and give my reference. So that he is very good scientist.
नीरज सिंह है वहाँ पर मैं.
नहीं, अभी just join किया. कल ही join किया.
सर GBFC नहीं सर, आप शायद GBU के बारे में बोल रहे हैं. GBFC में भी कोई नए साइंटिस्ट तो नहीं आया सर. कौन सा इंस्टीट्यूट? गुजरात बायोटेक्नोलॉजी रिसर्च सेंटर सर.
वही उसी में वो आए वहाँ से. हाँ, वहाँ पर
जबकि GBU में साइंटिस्ट थोड़े ज्वाइन होते हैं अनुपमा.
हाँ, वहाँ
फैकल्टी होंगे.
तो उनसे बात कर सकते हो.
पूनम मैडम
यस सर।
आपका कुछ क्वेश्चंस है?
नहीं सर, अभी तो क्वेश्चन नहीं है मेरा कोई भी।
पूनम मैडम ने इमटेक से सर किया पीएचडी एंड शी इज असिस्टेंट प्रोफेसर एट किरण पटेल ग्लोबल यूनिवर्सिटी बड़ौदा।
वेरी नाइस, नाइस टू मीट यू।
और बीएचयू से किया इन्होंने एमएससी।
ग्रेट, ठीक है। अब तो कोई ज्यादा क्वेश्चन नहीं है तो सर यू कैन एक्सप्लेन अबाउट द बीटी प्रेजेंट यूजेज।
सर विद्यानाथ झा सर, आप कुछ क्वेश्चन पूछना चाह रहे हैं?
एक वो पूछा है किसी ने कि वो बीटी के बारे में।
yes.
बीटी के बारे में जो क्वेश्चंस है जनरली अप्रेजेंस था कि ये बीटी जो है अब बहुत ज्यादा हमेशा whenever we talk about the GM crops people talk about BT is right now it is now being is basically it's a toxin or protoxin which is not at all toxin, and that is present in one bacterial Bacillus species, and people have taken the gene, modified the gene, and then transformed to the plants. And when a particular insect, that is a lepidopteran insect, when they chew, it goes in the gut of those insects, and there because of some of the peptides which or the proteases which are present in their gut, it turns from the protoxin to toxin, and this is not possible in the case of human or any other the mammalian or any other things. It's only with the one type of insect. So that becomes it actually interact with their some of the receptors which are present in the gut, which is not present in our system, and then it becomes toxic. So it's not only that it is because of that, it is the BT toxin is people have used all these things. Even whatever you in the Gujarat, if you are in the Gujarat, the people who are growing in the all these BT cotton, it is quite common in Gujarat. So I think that is also people have already doing all these things, and it has been growing since I think 2008 onwards. And still now it has spread up to near about 90% of the crop which are growing in the Gujarat. That is a BT cotton that is growing. So there the people are stopping. The major advantage is that people have stopped the using of the pesticides. Usually in the cotton you have to spray near about 8 to 10 times in their entire life cycle. So that they have reduced. So for the cotton ball they have reduced to 1 or 2 times only during the fruiting that now people are spreading. So this is we are actually having the less environmental impact in those things. That is my opinion.
Thank you, sir. Sir, Professor Vidyanatha Jha, do you have any questions, sir? Question पूछना है सर?
क्वेश्चन नहीं है, ऑब्जर्वेशन है. ऑब्जर्वेशन यदि आप चाहें तो मैं बोल सकता हूँ.
हाँ बिल्कुल.
जी सर.
ठीक है. सबसे पहले तो मैं आपको धन्यवाद देता हूँ कि यू आर कंटिन्यूइंग दिस स्प्री. आप निरंतर अपने काम में लगे हुए हैं. ये मार्के की बात है. और अपने अपने फील्ड में जो लोग डिस्टिंग्विश्ड जो भी अचीवमेंट लोगों ने किया है उनको इकट्ठा कर आप ये काम करवा रहे हैं रिलेंटलेसली. दैट फॉर दैट आई कंग्रेचुलेट यू. मेनी थैंक्स. और जहाँ तक जीएम क्रॉप की बात है, वी एंड आवर कंट्री इज पासिंग थ्रू ए फेज ऑफ ट्रांजिशन. तो नॉन एडिबल में तो सरकार को झंझट नहीं है, लोगों को भी झंझट नहीं है, लेकिन जो एडिबल प्लांट्स है, एडिबल क्रॉप्स है, उसको लेकर के झंझट है, संशय बरकरार है. तो इसके लिए आपको रिलेंटलेसली फाइट करना होगा. लोगों का जो ओपिनियन है, सरकार का जो ओपिनियन है, उसको मोल्ड करने के लिए आपको ये इस तरह से आपको ये लगातार अभियान चलाना पड़ेगा. आखिर नेचर ऑल्सो टुक थाउजेंड्स ऑफ ईयर्स इन डेवलपिंग ए न्यू वेराइटी एंड और समथिंग लाइक दैट. तो उसको तो लोग फिर एक्सेप्ट कर रहे हैं, लेकिन आज लेबोरेटरी में बहुत शॉर्ट ड्यूरेशन में आप नई चीजें बना रहे हैं, तो उसको एक्सेप्ट करने में थोड़ी झिझक हो रही है. तो नेचर अगेन, टाइम अगेन विल टेक इट्स ओन कोर्स. और इसको हम लोग बढ़ाते रहें, ये डिस्कोर्स चलता रहे, यही समय की मांग है। बहुत बहुत धन्यवाद।
Thank you very much, sir. Sir, Praveen Verma, sir, he is my teacher and served as a principal in MLSM college, MRM college, CM Science में भी थे, sir.
नमस्कार, नमस्कार सर।
नमस्कार, नमस्कार। बहुत बहुत धन्यवाद। आप इसी तरह से करते रहिए, हमारी शुभकामना है।
सर रेगुलर कर रहे हैं, तभी तो हम R&D के डायरेक्टर, जेएनयू में R&D सेल के सर डायरेक्टर थे। Yes, sir.
ठीक है,
I'm really grateful to Praveen Verma sir for joining us and delivering lecture on this platform.
Thank you.
Our all purpose is to disseminate quality knowledge, authentic knowledge, because we have observed that there are so many lakhs of students graduated every year in PG level or PhD level, but most of the major proportions they are not aware of about all the advanced tools and techniques, advanced biology, streams of biology. That's why we have started this, so that researchers and different MSc domain, the student will joining free. Sometimes I get learn and know, and I'm thankful heard by some of the participants. Just sir certificate कब मिलेगा? जबकि हमने इसी time में this Google form, so that we can come to know who has joined, and then I will give you certificate. Those who are not joining, they are also claiming that certificate कहाँ से मिलेगा? That is very very bad trend. लोग बहुत सारे registration से पहले ही पूछते हैं कि certificate मिलेगा कि नहीं. We have opened this for learning, discussion, so that हम लोग बच्चा थे तो विद्यानाथ झा सर ने हमको जेएनयू में कैसे एडमिशन लेना है वो लिख के दिया था कि क्वेश्चन कैसे मंगवाएंगे. And nowadays JNU R&D head and Oxford faculties and Cambridge and other, we are due to this online platform we are arranging those lectures, and students are and researchers are not joining, they are not taking benefit of these things. That is hurting. We are trying to improve overall knowledge pool in biology. हर सब्जेक्ट सर्वाइवल के लिए जूझ रहा है. लोग बोलते हैं बच्चे नहीं आ रहे हैं, और हम बोल रहे हैं कि जो बच्चे हैं बीएससी का बच्चा को बीएससी का फुल फॉर्म नहीं आ रहा है. बिकॉज आई विजिट टू सेवरल कॉलेजेस, अब वो भी बातें बोलेगा बायोटेक में कुछ नहीं हो रहा है, उसमें जो है ना कुछ स्कोप नहीं है. भैया आपको बीएससी का फुल फॉर्म नहीं आ रहा है, आपको डीएनए का फुल फॉर्म नहीं आ रहा है. यू हैव जस्ट परचेज द डिग्री एंड देन यू आर सेइंग कि नहीं हो रहा है. बहुत सारे लोग, ये भी एक सिस्टम का भी दोष है. बट मेरा है कि मैक्सिमम यूटिलाइजेशन हो रिसोर्सेज. देखिए अभी भी एक निधि ओझा, शी इज नाउ ज्वाइनिंग, शी हैज सेंड रिक्वेस्ट, खत्म हो गया फिर भी नाउ शी विल सेंड फोटो, सर हम तो ज्वाइन किए हुए थे, एंड देन दे नीड सर्टिफिकेट. सो विद दिस I will not go more. So thank you again, sir, and all the participants who are joining. That is also motivating for us, so that we are continuing this series, and it will be like a treasure for coming generation. All these good lectures are available, depending upon the consent of the speaker. We can either share PPT or this PDF or recorded lecture. So you all should keep in touch through my social sites. And again on 13th there is lecture on RT-PCR based diagnostic. So you all should join. So now Poonam madam.
गोपाल जी,
सर
जी लास्ट आप बोल के वाइंड अप कर देते. हाँ मैडम.
So one question, it may not be relevant to the topic. So what do you think about the scope of RNAi based biopesticides in future?
Oh, great! There's these people have already started in the Australian group, where the one Indian scientist, Dr. Neena Mittal, she's pioneer in that. And what recently she has delivered the lecture in my university and my school itself, and she has told that they are actually the major problem with the RNA based pesticides when the new generation PSD, the pesticides people are talking about. There are two major reasons. One is that how it is going to be sustained in the field whenever you are spraying. So it should not get degraded that fast. So that now they have added you just search for in the Google bioclay. Bioclay, you just search and you will find that they have got a material. It's a nanomaterial basically, where they have made it is much more important. Second thing was important for is the basically the when we talk about this RNAi based pesticide or microRNA based siRNA based pesticide. So there the important is that cost effectiveness. So now that what she is telling that the cost they have got reduced, and you require to have very less amount of that particular microRNA for spraying. So I think it is going to be a future. So some labs in India also, they have already started working on these lines. My lab is also working in this particular line, and we have already collaborated with them who has developed all these techniques.
We need high throughput field trials, right, sir? Will it possible in India?
Yeah, still in my group when people are getting this particular project. So some people from ICRISAT and Indian Agriculture Research Institute and one or two more institute they have got, and they do have a good field trial. They have already done the field trial, and it is actually working very well. What they find. So
Thank
I
you
think
so much.
field trial is not a big problem in this.
Thank you. So if there is no more question, you can suggest also, you can share your feedback also. Dr. Rita Mahapatra, are you here? Okay. So,
Yes, sir.
do you have any question, any feedback, madam?
Sir, it's very good to listen, Praveen sir, because I'm also a plant biotechnologist.
Can you introduce, madam?
Yes, sir. I'm working along with Dr. Poonam Mishra at Krishna's School of Science at KPGU, Vadodara. Yeah, I worked on maize plant against disease resistance, that is using fungal elicitors. Sir,
Good.
so it's my not just a question or a suggestion that actually we are using GMOs, but those are not actually getting certified from the government. So as I have seen in my crop, that is in maize, the elicitor which I have derived from the fungus is doing well in giving the high yield quality and also disease-free plants. So I want.
It's good thing that you can go for all these things. It's a good strategy. Many people have started doing all these elicitors, even salicylic acid spray. Many things, sustainable things people have already done in this particular field. Think about something new. In what I always tell to the researchers in India, that the elicitors has already been used, in several things, and we have to keep one thing in mind always, that it is the cost effectiveness. How costly because the farmers will only use when it will be cost effective. They will.
Yes, sir. Yes, sir. I have tested in field trials also. 4 years field trials we have done.
Great, very nice.
So it's working very good, and even farmers were also happy, and they are actually asking when you are taking it to market.
I think it should go immediately for all these things. Yeah.
So you can connect with Verma sir on LinkedIn. Those who wish to ask more question, and in future
you can write to me on my
Yes. Mail ID also.
Mail ID also.
Yes. Rita Mahapatra is also HOD at KGPU. वहाँ पर मैं वो माइक्रोबायोलॉजी की हेड हैं, सर.
सर बायोटेक्नोलॉजी हेड एट KPGU.
अच्छा, बायोटेक्नोलॉजी. So now,
Thank you, sir, for the acknowledgement. But we are very happy to listen you, sir. And also thank you, Gopal sir, that you are arranging such lectures for students, also for the faculties. That it is very good. Many of our students are joining you, and also they are asking us also that what about this subject, and they are growing actually.
सर अपॉर्चुनिटी पर मैं भी अच्छा लेक्चर लेते हैं प्रवीण कुमार वर्मा सर.
अभी एक क्वेश्चन आया था कि हाउ that in the chat box is there, but I can take another lecture.
हाँ बहुत टाइम भी हो गया, बहुत सारे participants. Yes.
So yeah, they wanted the career perspective, so I can give a very good lecture on these things.
Yes. So Poonam madam, can you wind up formally?
Yes, sir, sure. So I don't think that I need to just conclude this session, because whatever being the discussion after the lecture of sir, it has been very interactive kind of thing that all the participants, they have enjoyed probably, and there are many questions which have been asked to Praveen sir. So I think that they have all tried to understand, the scope of these GMOs and how these can be fruitful to be utilized in future. So Praveen sir, thank you so much for joining us in this Science Dialogue series.
My pleasure.
And again, thank you, Gopal sir.
I never say no to Dr. Gopal.
Yes.
Whenever I find time, I join him.
Thank you.
So,
Sir, actually, I would like to thank Gopal sir also. And in each this Science Dialogue series, I actually thank Gopal sir, because I don't know how much people appreciate or how many participants they are going to appreciate this kind of initiative. Because the thing is that students or participants, they are getting the knowledge from the experts, and this may not be available to them even after they are paying. If they may be paying hundreds or thousand rupees, even then they cannot get these kind of information from the experts. They have to join. If they are joining one kind of topic, they may be getting knowledge about it, but not so many, so they can join for each of the topic. But these lecture series is something like that every now and then, in probably the gap of 15 days, in every fortnight we have such kind of lecture from the different experts, and they are not just from India, some internationally acclaimed scientists, they have also joined us. They have delivered their lectures, very prominent scientists from the IVAB or some other very important institutes. And currently, sir, since you are, and what should I say, since I have done my PhD from MTech, and I have huge respect for JNU, being my own university, which has given us a PhD degree. So I have a high regard for you. And again, thank you, sir, Praveen sir, as well as Gopal Jee sir, for conducting or initiating. Basically, the initiation is very important, and then the execution for every 15 days or every 10 days, just as Gopal Jee sir, he has mentioned that on day after tomorrow, on 13th, again there is going to be a Science Dialogue series lecture. So
13th, 15th and 19th.
Yes, that is what. In every now and then we are having such kind of lecture. So this is making the awareness among the students also.
मैं जब केजी कर रहा था तो 2000 रुपया दे के कॉन्फ्रेंस में जाके ट्रैवलिंग खर्च करके और 10-12 लोगों को
सही बात है सर। ये मतलब जिस तरह का कंटेंट यहाँ पे डिलीवर हो रहा है ना, क्योंकि बात ये कि फ्रॉम द वेरी स्क्रैच टू द हाई पॉइंट, आप एक फेज के थ्रू हर लेक्चर जाता है। हर लेक्चर में शुरुआत बहुत बेसिक है, एंड एवरी टाइम द ओरेटर ही delve into the in the heights of that topic. They take that in very good way. That जो कनेक्शन है ना साइंस के साथ में, वो भी बनता है लोगों का, बच्चों का वो भी बनता है, तो they get admire that yes, this can be the potential. Because as far as I can remember, in my college there were few students who joined BSc Biotechnology, and they were not aware that what they are going to do in biotechnology. That was the situation, and we try to explain them, we try to tell them that what could be the potential of biotechnology in real terms. But nowadays, we are forwarding these kind of lecture series to our students. They are also joining. Few of them every now and then they are joining. The rate of joining is also increasing. So the thing is that they are also getting the knowledge from a professor from JNU, which is very good at this level. So I cannot thank more for you to this Gopalji sir, and again sir, Praveen sir, thank you so much for your nice talk, your nice interaction with the participants.
And
Thank you
even
so much,
after
sir.
retiring, Professor Vidyanath sir also
Yes,
joined
sir.
and appreciate me. नहीं तो रिटायर होने के बाद तो लर्निंग ट्रेजेक्टरी कम हो जाता है, लेकिन सर भी ज्वाइन करते हैं. So with this, yes, thank you everybody. Now, good night.
Thank you so much everyone. So sir, we will be closing this meet now.
Yes.
Okay sir. Okay.
Good night.